about allele-specific analysis of cis-regulatory architecture. Thanks.
Thank you. Wait, this is working? Should be. Yay! Don't need to do the next. So hi everybody, I'm Swann, and thanks for the organizing committee to allow me to present my work here, and thank you for being here. So I'm a PhD student in the lab at the Biology Institute of the École Normale Supérieure in Paris, and today I'm going to present to you my PhD work. So I'm interested in transcription regulation, as most of you are, and in the different regulatory layers that it's involving, such as chromatin accessibility, that prevents the transcription factor to bind or not an answer. You also have the histone modification landscape that will allow you to have some epigenetic information, but also the end gene expression level, also that can be of interest. And more specifically, what I'm interested in is how the genetic variation underlying within the DNA sequence can impact all those different regulatory layers. So usually when people want to assess the impact of a single nucleotide polymorphism on a given layer, transcriptional layer, what we usually perform is a quantitative trait loci analysis, trying to link a given genotype, so usually a SNP, to a given phenotype. And the phenotype is statistically found within a population with a segregating genotype. The phenotype can be fly color, but it also can be a different measure, so quantitative measures such as chromatin accessibility, histone modification, or gene expression, which is the most common, that is called EQTL. So as I say, this is for trying to link the SNP information for a given regulatory layer. But what I'm interested in more is how to not only assess the impact of a single layer, but assess the impact of genetic variation on the whole transcriptional regulation, so involving multiple layers altogether. So how do you do that? We try to enter that by using allele-specific data. And this has been done with a close collaboration with the Furlong laboratory at the EMBL, with two persons performing the wet lab part, Bing and Rebecca, and also David and Emily for the bioinformatic analysis. So why would allele-specific data be useful for trying to answer such question? Is, as already been partly explained by Professor Deplanck, because it's a matter of cis and trans effect. If, for example, you cross two Drosophila lines that are inbred, you can look at the mutation. For example, here you have parent B that has a mutation on the transcription factor binding site that prevents it from binding, and therefore you end up with a lower gene expression output for both alleles. And if you cross those two parents together, you end up with a heterozygous F1 that will bear both alleles and bear the mutation only at one of the two alleles. And this way, because it's a common cellular environment for the two alleles, you don't have any trans effect in the case, but you only have the cis effect caused by this SNP in the blue allele, and then you will see if you have allele-specific data, then you can measure the signal coming from each allele independently, and therefore you can see indeed that this mutation seems to be causing a decrease in gene expression. So we have this allele-specific data, and we can compute two different measures. We have the allelic ratio, which is computing the proportion of signal coming from one parent compared to the other. And we have just a total count signal by summing up the signal coming from both alleles. So as if you had non-allele-specific data. Okay, so we have allele-specific data for 4 different assays. We have a TAC-seq, 2 histone chip-seq, and 1 RNA-seq. We have performed those different experiments on 8 Drosophila lines. So it's homozygous parent. The same mother has been crossed to 8 different fathers from the Drosophila genetic reference panel. And finally, all those samples have been sampled at 3 different time points that are crucial windows of embryonic development. So with this peculiar data set, we are able to look at regulation of transcription through different axes, so throughout different regulatory layers, throughout different genetic background, but also throughout different time development. So this is the kind of data set I'm using, or I'm working with. Here you have the example of a genome region next to the SCAP gene, and you have the 4 different assay signal that is allele-specific, and you have the maternal track at the top and the paternal track at the bottom. And you can see here that in this specific region, you are maternally biased for all the different regulatory layers, because more signal is coming from the maternal track. Okay, so back to the question that I presented to you before. How using allele-specific data will help us to try to understand the correlation between all these layers? We will use this data and try to correlate them among the different regulatory layers. By correlating the different regulatory layers, what I mean is we will try to link those different assays in a location-wise. So we assign, for example, an attack peak to a given gene if they're closer than 500 base pair together. And once you're assigning your different features together, you can just take the allele-specific data you have for each of them. So in this story example, you have a high chromatin accessibility for both alleles, but you have one of the 2 genes that have a low throughput in gene expression. And then with this data, you can try 2 different ways of correlations. First, you have the correlation on total signal, so non-allele specific, by just summing up the signal from both alleles. But you can also correlate the allelic ratio, in fact. So you lose the information on total signal, but you gain the information about bias and imbalance. So for example, in that case, you will see that the chromatin accessibility is balanced, but the gene expression level is imbalanced toward the orange gene. Okay? So when I speak about correlation, it's trying to assess the proportion of shared variance between 2 different variables. But usually what we want to get rid of in those specific cases is the potential confounding factors. So actually we want the true direct correlation between 2 variables that is not caused by an indirect factor, such as, for example, gender in a study. You don't want to take the indirect correlation caused by other variables. So in our case, for example, if you want to take the correlation between RNA and attack, the confounding factors that you could potentially have in your data set that could bias the correlation would be the histone mark. So how do we get rid of that? We use the partial correlation analysis. So to briefly explain it to you, you start from 2 variables. So here we have a significant correlation between our allelic ratio in RNA and attack. You take those 2 variables and you make a linear correlation with the other confounding factor. So in our case, as I say, it's the 2 histone mark. You correlate them independently, and what you do is that you extract from those 2 regression the residuals, so actually the variation that is not explained by those histone mark, and you correlate them together. And in that case, you have the direct correlation between the RNA and the attack that is not explained by the histone variance. Okay? So I've done that on the 2 measures I explained you just before, the total count and the allelic ratio. Just to present you first the result from the total signal, we get something like this. So here you have the 4 layers showed, and the line between the layer corresponds to the correlation. The thicker the layer, the higher the correlation. And if there is no line, it's not significant. And in this case, you can see that looking at just the total signal, so non-allele specific, you have a nice correlation between the 2 histone marks, so H3K27 acetyl and H3K4ME3. And you also have a nice correlation between A3K27 acetyl and the RNA. But you don't have such a strong correlation for the RNA and the K4ME3. And this kind of study has already been performed by the Vingron lab in 2013 with a much bigger number of histone data. But if we burn down that network to the same data set that we have, they kind of get the same structure as we end up having, with a strong signal between the 2 histone marks, a signal between the K27 and the RNA, but no apparent direct correlation between the K4ME3 and the RNA. So it was a good control. And something we can go further with this kind of data is, because it's allele-specific, we can do the exact same analysis, but this time not using total count signal, but the allelic ratio signal. And in that case, we get this kind of structure. So again, correlation is shown by the lines between the S's. So it's quite different from the one we found from the total count correlation. And if I put them both side by side, you can clearly see that there is a different structure between the two, with the allelic ratio completely losing the highest correlation we saw between the two histone mark, and also the correlation we saw with the K27 acid mark. However, we win a very strong direct correlation between the RNA and the K4ME3. So all that was for the features that are close to a transcriptional start site. But we also performed that for the features that are distal from those gene start site. But in that case, we don't have such a big difference in the structure. So it's really about the TSS proximal location. And it seems like the histone mark seems to play a different role in the correlation with RNA, in the propagation of total signal or allelic ratio. Seems to be a different action on those histone marks. So we wanted to go a bit deeper into what we gain from Allele-specific data. We also look at the enrichment for gene categories for relating to allelic imbalance. And you can see the redder, the higher the category is likely to be imbalanced. In contrary, the more blue, the less likely it is. And you can see that the gene categories that seems to be more enriched in allelic imbalance are unclassified gene or metabolism-related genes. Unclassified genes being the genes that are Drosophila specific, so highly evolving. And also in the blue categories, you can see that it's mainly transcription factor related, which makes sense because you expect those main regulators to be more sensible to change in regulation, so maybe they're more buffered in allelic imbalance. Yep. So just for the take-home messages about that, the Allele-specific data is a very useful tool to try to look at cis effects and trying at the same time to minimize all the potential trans effects you can have in your dataset. And that using partial correlation analysis is a very useful application also to try to get rid of indirect correlation. So you can measure actually direct correlation between two variables without other potential confounding factors. And finally, that using those two, both Allele-specific data and partial correlation analysis, we end up looking at the regulation, and it might look like the histones signal seems to play a different role in the correlation between allelic ratio and total signal. So something to dig a bit more. So just to thank all the people that have been involved in such work. So I would like to thank the Furlong Lab, also the CSB Lab hosting me as a PhD student, especially the big thank for the people that have been involved in putting the nuts and gear of the dataset, the project team, and the thesis committee member, and you for your attention. I'll be happy to take questions.
Yeah, hi. I mean, this is, I don't know whether this might be a stupid question, but why do you see this big imbalance, this big difference between the allelic imbalance and the just, yeah, exactly here? So what's your explanation, or why is that so different?
So it's kind of like it's hard to have a proof answer with some proof, but I might thought maybe some signal is more likely to act on both alleles, whether some other histone mark maybe are really allele specific and you can hardly have a kind of transvection to the other alleles, but it's open to hypothesis. And what is most striking to me is that in the total count correlation, you don't have a correlation between the K4ME3 and the RNA, which is kind of something that we really would expect to see. But
I mean, can you from this infer where would be a third actor, like a third factor interacting? At which edges would it be intervening? At which nodes would it be your whatever latent factor that you're missing here? Can you predict that from your data?
Ooh. I don't know. Well, the thing is that if there is no correlation already here, having one more factor wouldn't create correlation out of the blue. That is something that is already not present. So here, the loss of correlation is something that is set. But for example, all the correlation we can see here might be still indirect if we bring more variables and more histone mark together in the data set. So yeah.
So I had the same question. I have maybe some ideas. So the difference at the TS proximal sites could be that you have subsets of genes where K4 trimethylation correlates really nicely with transcriptions, and other it doesn't. And so when you look at allelic differences, you compare the same gene under different conditions, and so therefore you see the difference. Could there be? So have you looked into the subset of genes that show the strong correlation, whether they fall into a certain group?
So
Housekeeping versus developmental genes, for example.
there is no striking category that shows up, but also just this is partial correlation, so it's taking only the residuals from the other correlation between the two confounding factors. And if you just look at simple standard correlation, all the layers are actually well correlated between each other. So it's something that really taking, like, it's not the main variance that is explaining, it's explaining those partial correlation analysis here. Making sense?
So what is not the main variable?
So here, like, partial correlation is correlating the residuals. So actually, if you look at the standard correlation between the layers, it's already quite high, and it's the case for all the different pairwise correlation between the different layers. So it's pretty hard to look at the more correlated genes. But I don't see any categorical enrichment specific.
Okay, well, we can talk about it later. And so the other question, I think you didn't mention specifically, but it seems like the distal and the proximal difference should be very, I mean, that's consistent with the literature, right? So you have K4 trimethylation more at promoters than it enhances. So that would exactly be what you would expect, right?
Yeah, like for the TSS distal, we didn't look at the K4 signal because for partial correlation, you need to have value at all the features to make the correlation.
Okay.
And because K4 emi3 is actually mostly at the promoter, we ignored it for the TSS distal analysis because we couldn't have this data.
Okay, maybe I didn't understand your analysis. Let's talk later.
Sure.
Quick question.
So when you're doing this partial correlation analysis, there's some kind of hierarchy, right? First you take a correlation, then you regress it out, and then you look at the residuals. So how did you decide that hierarchy in which to do the partial correlation analysis?
So it's based on what kind of variables you have. It's the step when you regress out the residuals is you take any potential confounding factors. So actually all the variables you have that are not already within your pairwise correlation.
Okay, but like, so then if you said everything was well correlated, that would mean that if you had regressed out K4ME3, then you would probably get poor partial correlation between K27 acetyl and RNA, right? Because if everything is well correlated.
It's already well correlated, but you still have this. So I wouldn't bet that you'd lose everything, but there are still residuals coming in. It's just overall the correlation is quite well, even without that.
Okay, interesting. So could that be maybe like the other variables are maybe just noisier? So like even when you regress them out, there's still signal for like a particular pair?
It can either be that, or either that we don't have, there is another variable coming in that we don't have the measure for, and that is bringing this additional variance. So something we cannot test, really.
Thanks, Swann, again.
Thank you.